Hello, everybody. I am Rodolphe Thiebaud. I'm professor in public health and biostatistics at Bordeaux University in France. And it is my great pleasure to welcome you to this seminar about design and analysis of COVID vaccine trials. It has been organized by the scientific committee in charge of the 9th Statistics in Biopharmacy conference to be held in Paris on 29 to 21st of September this 2022. And so keep in mind that the abstract for this conference will open in mid-January and close at the end of March. So now let's enjoy this webinar with two great invited speakers. Dr. Holly Janes, she's a biostatistician working on the design and analysis of vaccine studies, and she's co-PI of the Statistics and Data Management Center of the HIV Vaccine Trial Networks in Seattle with Peter Gilbert. And Stephen Senn is a statistician specialized in statistical methods for drug development, who wrote a fantastic book. This one, I'm sure he's going to talk about it. And I'm still waiting for the dedication, Stephen, so hope I will get it. So after those two presentations of 30 minutes each, we'll have 30 minutes for discussion. So questions are welcome. Do not hesitate to ask your questions. You get a specific box on the right with questions. So you just ask your questions, and we may be able to answer these questions either directly by writing or just by raising your questions. We will not be able to give you the mics because we get more than 150 people who registered to this webinar. Okay, so now I guess we can start. So Stephen, if you want to share your presentation, the floor is yours, and you will be able to start.
Okay, can you see my presentation?
We see it very well. Thank you, Stephen.
Good, okay. Okay, so thanks for the invitation. I'll crack on with the presentation. So I'm going to talk about infectious processes, then I'm going to have a look at 5 vaccine trials. Then I'm going to pay some attention to one of them, the AstraZeneca Oxford study, and then have a look at another, the Pfizer study, try and draw together some general points from these case histories, if you like, and offer you some conclusions. And this is actually taken from the extra COVID chapter that I wrote for a second edition of Dicing with Death, which should appear next year. So here is a toy epidemic that I've simulated. I've taken a population of only 100 persons. I've assumed that there are 2 initial introductions. I've assumed that everything works in a sort of discrete time. So you have period number 1, period number 2, period number 3. Obviously, that's unrealistic, but it's just a simple way to illustrate some points. And I have taken this famous R0 number to be equal to 0.8. The important thing here is it's less than 1. That means that on average, each case infects fewer than 1 individual. And so eventually the epidemic effectively fails to take off. On the left-hand side, you see the number of cases infected in any given period. The probability limits, they're not confidence limits because we're not talking about a parameter here. The probability limits for 5% and 2.5% and 97.5%, the quantiles are given. And also given is the median line, which you can see rapidly goes down towards 0. So this particular epidemic, in fact, extinguishes itself. Of course, individual traces are given in blue, so some epidemics may take off, but typically you'll find that only 3 or 4 cases occur in the population altogether. On the right-hand side, you have the cumulative effect of this particular epidemic, so the total number of persons who've been infected, and you can see that for most of the epidemics, it doesn't even reach 30 out of the 100. Now, all we need to do is to use the reciprocal of the value R here, so going from 0.8 to 1.25, and we then get a very different situation. Now I've simulated using a value of R0 equals 1.25, and you can see we get far more cases period by period, irrespective of which particular simulation we're looking at. I've simulated 500 times. And on the right-hand side, you can see the total number of infected, and the typical number is much higher now, and quite easily we can get to a situation where 65 to 70% of the population are infected. So just a simple difference in R naught, a small difference in infectivity, if we're round about the critical point, can make a big difference. And that's one of the motives for finding effective vaccines, is the hope that we will also contribute to reducing transmission, not just protecting the individuals who are vaccinated. So I'm now going to have a look at 5 vaccine programs. They are the Pfizer-BioNTech program, the AstraZeneca-Oxford one, the Moderna one, the Novavax one, and the J&J Janssen. I myself have had the 2 initial AstraZeneca-Oxford jabs, and here in Scotland we would say JAG, not jab, and I've also had a Moderna booster fairly recently. So I'm three times I've been injected. On the left-hand side, you can see the boundaries that there were for 4 of the trials. J&J Janssen used a sequential probability test, a continuous monitoring, so that the boundary is rather different, difficult to represent on this particular diagram and difficult to find from their protocol. But you can see that they're looking at very, very similar design parameters. 5 of the sponsors assumed that the vaccine had to have at least 30% efficacy to be of interest. And under the alternative hypothesis, 4 of them assumed 60%, the exception being Novavax, who assumed 70%. They planned for power of 90 to 95%, and the sort of events they were targeting were between 100 for Novavax, but remember, they assumed a rather larger value under H1, to 164 for Pfizer. And you can see the number of subjects that were targeted, but the number of subjects you have to target depends upon your background assumption about infectivity. So a surprisingly effective analysis, which I shall show you in due course, just uses the total number of cases. In fact, a useful parameter is this relative infection rate, if one might call it that way, which is theta, which is simply the ratio of the probability of being infected if you're given vaccine to the sum of the 2 probabilities, the sum under vaccine and the sum under placebo. And it can be estimated in the way I've shown on the slide here. And if it's the case that the numbers of subjects in the 2 arms are approximately equal, effectively it reduces to an analysis which is based upon the number of cases only. And in fact, during the various blogs, for the various blogs I wrote during the epidemic, when I didn't have much information, I simply calculated using the number of cases which you could generally get from the press releases. You can transform this to the vaccine efficacy case. The vaccine efficacy scale is the reduction for vaccine compared to placebo divided by the placebo rate. So if we have a look at the results of the 5 large vaccine trials, those are the subjects that were treated in the first 2 columns, and then in the columns 3 and 4, you have the number of cases. What you've got here in the diagram on the right is the point estimate and the confidence or credible interval in the case of Pfizer, as calculated by the company, represented by a thick, solid blue point and solid black lines. Underneath, I've done a simple analysis using the cases only. I've not used the number of subjects. I've not used any covariate information. I've just used a simple exact binomial analysis. In the case of AstraZeneca Oxford, I had to adjust this because they used a 2-to-1 ratio. If you have a look at the AstraZeneca numbers, you'll see that they were in fact recruiting twice as many vaccine subjects as placebo subjects. A rather unwise decision, in my opinion. I'll explain that in due course. But you can see, if you look at the right-hand side, that my simple analysis got remarkably close, improbably close. I was surprised how close it got to the more sophisticated analyses that the companies were prepared to produce. By the way, you should always prefer their analysis to mine. They are the experts, not me. Their analysis pre-specified, they had more information. If we look at the observed allocation ratios for the trial, then the planned allocation ratios were 1 in every case except for AstraZeneca Oxford, which was 2. And the observed allocation ratios are given there as well. And I've also done a little chi-square test to see to what extent the allocation ratio differs from what was predicted or expected. And the chi-square is in fact less than 1 in every case. Not surprising, because they will have used permuted blocks to balance the numbers, and I'm just using a simple binomial assumption here. So the variation will be less than the binomial. But the surprising thing is that the AstraZeneca chi-square is 6.025. The discrepancy is 6 times what you would expect on the chi-square scale, despite the fact that they would have been balancing using permuted blocks, I suspect. So a slight issue here, which I'm not aware has been addressed in any of the papers by AstraZeneca Oxford. If it has, please let me know. I'd be interested to see what they have to say. So I'm now going to look at the AstraZeneca Oxford program in some detail. This is a diagram which you can regard as representing the plan, if you like. So they assumed a particular baseline rate for persons who would develop COVID if they were unvaccinated, and clearly that's the value for the placebo group. And then, of course, they would assume a particular vaccine efficacy, 30% under the null hypothesis and 60% under the alternative hypothesis, to generate the number of cases they would expect to see. Remember, we're talking about planning here, in the vaccine group. And so here what I've got is I've got two contours, probability of bivariate probability contours, sets of contours. The right-hand one is what applies in the case of the null hypothesis when we have 30% efficacy. And the left-hand one is the one that applies for the alternative hypothesis when we have 60% efficacy. And various lines that I've drawn sloping from bottom left to top right correspond to particular levels of vaccine efficacy. For example, the 30% vaccine efficacy line, the yellow line, goes right through the center of the contours for the H0 distribution, whereas the 60% line goes right through the contours for the 60% power assumption. In between the two is a boundary, which is actually the boundary for significance. If the cases fall on that particular boundary, you would declare significance. So this is a sort of geometric distribution representation I quite like, which shows essentially everything. The red line is the boundary for significance. It's the white line is the boundary for 50% efficacy. Okay. Here's some trial simulations to see how this would work out. I've simulated using the design parameters that AstraZeneca used. On the left-hand side, we have the situation under H0. And on the right-hand side, I've added what the epidemic would look like. I've done a number of simulations here if, in fact, the alternative hypothesis of 60% vaccine efficacy applied. And the gratifying thing here is that there is strong separation between the red lines, which are the lines if the vaccine has the desired efficacy, and the blue lines if it is just ineffective. In other words, if the efficacy is 30%, and you will see there is a pretty good separation between the red lines and the blue lines. And of course, as statisticians, when we do a power calculation, this is essentially what we're trying to do. We're trying to show that what will happen if our alternative hypothesis is true will be rather different from what will happen if the null hypothesis is true, and therefore that we will be able to recognize which is which. Oxford's AZ Oxford's 2-to-1 allocation was not a wise choice, in my opinion. I calculate that if the 60% efficacy applied, it would increase the duration of their trial by 1/6. These particular lines here show the time it takes to complete the trial as a ratio of what would happen for a 1-to-1 allocation, depending upon particular efficacies that might apply. Here, the vaccine efficacy scale is a proportional. I've not multiplied by 100. But for a vaccine efficacy of 0.6, which is to say 60%, then in that case, you would get 1.167 times the duration that you would get if you had used a 1-to-1 allocation. So AstraZeneca inevitably put sand in the wheels by doing this. They delayed the point at which they were going to be able to say something meaningful about the vaccine. I can think of various arguments why they might want to do this, but I don't think these arguments are really that important compared to the business of getting the trial finished as early as possible. In fact, when they registered in the United Kingdom, they did not register based on this particular trial. They registered based on 2 other trials, neither of which had completed, both of which were analyzed ad interim, one of which had had a serious mistake in dosing. This is why you see here the labels SD for standard dose and LD for low dose. What you found is that some subjects were given the low dose, but the amazing thing is the efficacy seemed to be greater for the lower dose. That's something we generally don't expect in drug development. And the explanation was that perhaps due to the mistake, there had been a longer duration before the second dose was being given. And actually, it was the duration that was important. A longer duration between doses gave more efficacy. Bear in mind here that efficacy is judged from the second dose having been given. That's another point one could discuss. Ignore the diagram on the left, which gives you a number of cases. On the right, here is some evidence from spike binding antibodies that, in fact, the dosing interval was protecting subjects better. What we have here is that the black points and the accompanying confidence intervals tell you what the antibody levels were as a function of the interval between the first and the second AstraZeneca dose. Now, one might argue, well, it could be that different patients are involved, and certainly different patients would be involved, and this is not a randomized comparison. But that's where the red and the blue points come in. The blue points are the same spike binding antibodies, but at baseline. They cannot have been affected by the interval because they were measured before the interval, and they're more or less flat. Also, the red ones are the values after the first injection was given. They again cannot be affected by the interval because the interval is determined by the second injection, and they again are flat. But the black ones, which can have been affected by the interval, are not flat. So this is circumstantial evidence that, in fact, the interval was important in providing greater efficacy. These are a comparison results by interval. This is an example of a mean plot, such as you can find in Jason Hsu's very nice book. On the x-axis, you have every treatment. Here, the treatment is the interval. And on the y-axis, you have every treatment. Here again, the treatment is the interval. And so you can plot any paired comparison using a point. And you can also have these diagonal lines, which effectively represent the confidence intervals. And the main diagonal going from bottom left to top right is a way to judge the significance or not. And you'll see that only the comparison of the longest interval to the shortest interval is significant on the left-hand side. It's still significant if I also add a Bonferroni correction. So now let's have a look at Pfizer-BioNTech. I'm now going to switch back, and I'm not going to use the most recent numbers of cases because they use a Bayesian analysis, and it's actually interesting to see what happens to the Bayesian analysis when we don't have too much information. When we have a large amount of information, then in that case, any prior distribution is overwhelmed. And so I thought it'd be more interesting to go back and have a look at what the situation was in December 2020, rather than the situation by the end of summer 2021. So Pfizer had a rather strange choice of prior distribution. They used a beta. That's not strange because they were actually doing a prior distribution for this theta parameter I talked about before. But they chose the parameters 0.700102 for the first beta parameter and 1 for the second one. Now that is 6 significant figures. 6 significant figures for a prior distribution, which itself is supposed to be rather uninformative. They're being incredibly precise about something itself, which is rather uninformative. This is really weird. And the reason is it transforms to 0.4118 as the mean prior value of theta, which when you transform using vaccine efficacy, transfers to 30% on vaccine efficacy scale. But this, in fact, is not logical for various reasons. I've also plotted the same prior distribution using the vaccine efficacy scale. By the way, note that vaccine efficacy can go down to -infinity. It's a very peculiar measure. The top value is 100%, but the bottom value is -infinity. It's another talk. These are the posterior distributions I get now for the theta parameter. And you can see I've chosen a number of possible I've chosen a number of possible other beta parameters, beta priors there. It doesn't really make much difference to the posterior here. And on the right-hand panel, you've got the actual posterior distribution that Pfizer ended up using. But the choice of prior is strange. The mean of the beta distribution is given by nu over nu plus omega, if those are the two parameters. So many choices are possible. What I have got here is I've got contours on the right-hand side. I've got contours of the variance of the beta distribution as a function of the two parameters. The particular parameter contribution that Pfizer chose is the one which has got the red point on it. But that's not the same as a variance of 1/12, which is the standard variance you get from a uniform prior. So even if they wanted to tweak the uniform prior to give it a different mean rather than the mean of 0.5, then in that case, it's a strange choice. They could have chosen a combination of parameters which would have given a variance of 1/12. So various possibilities are illustrated here. It's really not obvious to me why Pfizer did this. It strikes me as being a lot of complicated, a lot of sort of mathematical complication for no good reason. The statisticians were having fun one day and they thought, well, why not write this down and see what happens, You can, in fact, calculate confidence intervals in the frequentist way. What I have on the left-hand side is what the tail area binomial probability looks like for various values of theta. But on the right-hand side, I've got two curves of theta, two curves of tail area probabilities. One is the probability of 8 or fewer cases. Remember, there were 8 cases seen here, 8 cases in vaccine and 162 under placebo, or 8 or more. And so I can use each of these and pick off the tail area probability corresponding to 0.025. And I then have my 2 points of the 95% confidence interval, and they are given here. And the confidence interval on the vaccine efficacy scale goes from 90.04 to 97.81 with a vaccine efficacy of 95.06. This is my frequentist analysis, just using the cases and nothing else. Just using the fact that there were 8 cases on vaccine and 162 on placebo. So putting all that together, on the very top line, you have what Pfizer-BioNTech did with their full model for the credible interval. Point estimate of 95%, lower limit 90.3, upper limit 97.6. On the bottom line, you have my simple confidence interval analysis. The point estimate is 95%. And my limits are 90% to 97.9%. Very, very similar to what Pfizer got, although I'm using a frequentist method. In the middle, you have 3 possible point estimates that I did using my Bayesian analysis, either using the credible mean, the credible mode, or the credible median, and then using the credible limits. And you can see that my credible limits using cases only get remarkably close to what Pfizer used using all the information they had at their fingertips. I find this really quite astonishing that one can get so close with such a simple analysis. So some general design issues. First of all, the value of concurrent control. The actual infection rates varied enormously from trial to trial, and that's shown in the left-hand diagram, which for 3 of the studies shows you the vaccine incidence per 100,000 person years and the same for control. And you can see that these values vary very differently. But if you compare like with like, you get a fair comparison, and this is the value of concurrent control. What I've done on the right-hand side is I've played around in which I've compared, for example, AstraZeneca Oxford to its own control, and then said, well, supposing the values had been the same as in the Moderna trial under control, or supposing they'd been the same as in the Pfizer-BioNTech in control, what would it look like? And you get rather different answers. You can see that had Moderna control cases been the control for the AstraZeneca Oxford vaccine, you wouldn't even have assumed it was efficacious because the infection rate was so much lower. Why am I telling you this? I'm telling you this, we all know this. We all know the value of concurrent control. But many of the other pieces of information that we have got during the pandemic are of necessity not from clinical trials, and they therefore potentially suffer from all of these biases. So we should be aware of that. The value of blinding, I don't want to say too much about this, but just to say there are many ways in which running a clinical trial blind prevents other problems arising in the clinical trial. It's not just a question of judging the outcome. It's a question of making sure all sorts of things don't happen. For example, you might be tempted, if you don't run it double blind, to say, well, you know, we don't have to bring the control cases in to be vaccinated. What's the point? So we'll just bring the vaccine cases in, the treatment cases in to be vaccinated. But then in that case, of course, they all attend clinics at the same time. So this is potentially a loss of independence, and all sorts of other biasing factors can intervene. So you have to be very, very careful about trials which are not blinded by design. Here I've shown how autocorrelation could lead to clustering. I've put some autocorrelation into the effects seen in patients. I've just generated this from a particular distribution with an AR1 process. And on the left-hand side, I have a randomized trial where right throughout this process going on, right throughout, control and vaccine cases are scattered right throughout. But if what I do is I decide, right, I'm going to bring some cases to be vaccinated in, and then I'm going to deal with some control cases and so forth, then you get clustering of vaccine and control subjects according to the particular period in which the autocorrelation is having its effect. And so on the right-hand side, you find that you get potentially a biasing or a lack of independence resulting from the particular process. What policy for vaccination? Well, there's an issue as to when you should time the secondary jab, I suppose. What I have on the very top with the dotted line is the protection you would get if you had no vaccine at all. The red line tells you what happens when you have your first jab. And then at a particular point, what you could do is you could either have a second jab, say at 4 weeks or at 12 weeks. Now, what happens is eventually, if you look at the red curve, which is what will happen once you've been jabbed at 12 weeks, eventually you'll get more protection than you would get if you had been jabbed at 4 weeks. But of course, the period from 4 weeks to 12 weeks is an 8-week period in which you will not have had your second jab if you have the policy in which you have a longer duration between the two jabs. So this is a public health decision. It's a difficult decision to know, even if you suspect that the efficacy of the vaccine may be increased by increasing the interval between jabs. It doesn't mean that that particular policy will make the biggest contribution to reducing the infection rate, because there will be a longer period in which subjects have not received their second jab. Conclusions. So lessons. I think some good things were done. I honestly think that the life scientists did brilliant work. Statisticians also did good work, but we shouldn't kid ourselves. The really, really important work was done by the life sciences. The big trials were done rapidly. That's where the clinical research associates showed what good professional attitudes, training, and competence can do. Amazing the way these trials were run. The regulators worked fast, and the subsequent rollout was impressive. So all of that logistics stuff was extremely impressive. I offer for discussion Pfizer's bizarre choice of prior distribution. Didn't matter, but it was sort of really odd in my opinion. Oxford AstraZeneca's unwise choice of allocation ratio, that mattered somewhat. Not so much, but it was an unwise decision in my opinion. The interesting statistical finding that an analysis based on cases only gets remarkably close to what much more sophisticated analyses produce. And that might at least be useful as a way of doing a sort of back of the envelope check of various analyses when they're produced. The importance of current control and the importance of using an appropriate scale. I didn't really go into that, but that's also an important feature. So thank you very much.
Thank you very much. Thank you for this very interesting talk. I'm not seeing any clarification questions. I don't know whether there is any clarification question from the scientific committee member. I've got just one, Stefan. It's in your last slide, not the conclusion one, but the estimates of the hazard according to the duration between the first and the second jab. So these estimates are based on the data you get from
these were
just fake data.
These were plausible values. I was just trying to illustrate the issue. Obviously, to come to a decision, you would have to have some way of actually estimating the parameters. But I'm just showing there's an issue here. The issue is long-term protection versus, the short-term loss.
The message was clear, but I was wondering.
No, you're quite right to raise that question. I should have made that clear. Yeah.
Thank you.
They look too good. They look too good to be true.
Yeah, that's true. Okay, so now, Oli, are you ready to give your talk? And then afterwards, we'll have a general discussion coming back to Stefan's and your presentation. Okay, so we can see your slides very well. So the floor is yours, Oli. Thank you very much.
Thank you, and thank you, Stefan, for setting this up. I'm going to give some a background and information and commentary given my role in the COVID prevention network. So I'm a statistician. I sit at the Fred Hutch in Seattle, and many of us who worked in NIH-funded research related to vaccine development were immediately pivoted to working on these COVID vaccine trials that were U.S. government funded, and a new organization was formed called the COVID Prevention Network, which basically is an amalgam of everybody who was working in HIV and other pathogens with key experience in vaccine design and evaluation. Was sort of glued together in this newly formed global network to support the design, analysis, and conduct of the U.S. government-funded COVID vaccine trials, and there, the part of the structure here was a common laboratory group and a common statistical and data management group, with leadership played by Dean Fullman, who sits at the NIH and in the NIAID division, and Peter Gilbert, who sits with me at the Fred Hutch. And so this is really reflecting, our experience collectively over the past 18 months to 2 years in setting up and conducting these trials, which of course are still underway, and many studies still to be done, analyses still to be done, lessons still to be learned. So what I'm going to go through is some brief commentary on the rationale for the phase 3 trial designs that were conducted under the CoVPN. These were the phase 3 studies that received some form of U.S. government funding, which is not an exhaustive list of phase 3 trials that have been conducted to date. But these are the ones that I'm most familiar with and have some background on. I'll discuss how these designs were adaptive to the emerging data, in particular with rapid vaccination of the originally randomized placebo recipients, and lead into some open questions that remain and challenges in addressing these questions, given where we are in the pandemic today. So this is a high-level snapshot of the phase 3 trials that were and are being conducted under the auspices of the CoVPN. And a key principle of this program was to study a diversity of vaccine platforms, ranging from the novel mRNA vaccine platform to more established vaccine platforms, including adenoviral vector-based vaccines in the middle there, and protein-based vaccines at the bottom. Of course, the first of these studies to report out was the Moderna mRNA vaccine, demonstrating considerable and surprisingly dramatically high levels of efficacy with a vaccine that received emergency use authorization way back in December 2020, now fully licensed. The Janssen vaccine, an adenoviral vector-based vaccine, received emergency use authorization back in February 2021. Two other trials that have reported out, the AstraZeneca trial, as Stefan mentioned, which has a number of other studies that were not U.S. government funded, but the U.S. government funded study has resulted and and as has the Novavax study, but those vaccines are still under consideration by the US FDA. And then the Sanofi trial is the fifth in this series that is still underway with no results reported as of yet. So this is a high-level snapshot. We'll dig into these trials in more specific terms. But I wanted to highlight some key principles under this vaccine development program. The first was the notion of conducting a set of what were called harmonized phase 3 trial designs. So early on, there was considerable discussion as to how to evaluate in an efficient and rigorous way these 5 different vaccine platforms, and some thought put into whether or not a platform trial, putting all these vaccines head to head in a single trial would be best, but ultimately what was decided is that for expediency, it would be more feasible to conduct a series of harmonized phase 3 trial designs, which were conducted in parallel. We're looking back, I think this was the smart choice. There was considerable delay in the availability of some of these products. As many of us know, the process in getting agreement with multiple manufacturers to conduct a single trial is considerable and would have delayed the initiation of the trial considerably. So instead, what was done is really for this common statistical group to put together a common protocol and a statistical framework, and to a large extent, that common framework was applied to each of these trials with considerable collaboration with the industry partners, and that resulted in largely consistent protocols, common primary and secondary endpoints across the trials. A common data safety monitoring board was set up to have insight and oversight of all these trials in parallel. Common immunological laboratories to generate data that would be comparable across the trials. And then, of course, this common statistical group, which was, really an amalgam of statisticians at the Fred Hutch, at NIAID, and a number of other academic centers throughout the U.S., and groundwork laid for data sharing to permit cross-protocol analyses. So briefly, and I'll go through this pretty quickly because Stefan has commented on a number of these design elements. These are kind of the key design elements as I see them across these phase 3 trial designs, and they're common. They're common across these trial designs. Generally speaking, the trials were sized to be quite large, and I'll talk about the rationale for the sample sizes in a moment. A consideration was whether or not there should be pre-screening for individuals with prior infection. It was thought and known at the time that individuals with prior SARS-CoV-2 infection were at dramatically reduced risk of infection. It was unknown, what the risk of reinfection was, but the likely benefit of vaccination was certainly greater in individuals who were not previously exposed and infected to the pathogen. Despite this, a decision was made to recruit broadly, to not pre-screen trial participants for prior exposure or infection, in order to generate both safety and efficacy data for a broad at-risk population and enable licensure of vaccines for a broad at-risk population, not requiring pre-testing. The trials were enriched for individuals that were thought to be at high risk of poor outcomes associated with COVID-19 disease based on age, comorbidities, race, ethnicity, and a key consideration, especially in the U.S., given the disproportionate burden of COVID in underrepresented populations, was really a mandate to enroll minorities at or above their U.S. demographic frequencies. As Stefan mentioned, these trials, some used a 2-to-1 randomization ratio, some used a 1-to-1 randomization ratio, and there was considerable thought put into that with a variety of considerations around both statistical efficiency and precision, which can be a bit more precise under high vaccine efficacy with a 2-to-1 ratio, but more importantly, the 2-to-1 ratio permitting more powerful studies of immune correlates, which I'll talk about at the end of the slides. And as well, I think in retrospect, a potential advantage of the 2-to-1 randomization, that essentially the placebo arms for some of these trials were effectively lost over time. And so having randomized individuals 2-to-1 to vaccine or placebo actually has yielded more data in the long term on the effects of the vaccine. The trials were all planned to follow participants for 2 years post last vaccination with groups of sequential interim monitoring to allow the results to be reported early in the event that potential harm in efficacy or efficacy had been established. And the primary endpoint was virologically confirmed symptomatic COVID-19 disease. So to comment on that primary endpoint, first here's a quick snapshot of the specimen collection that was implemented. There's some variation across the trial designs. This is kind of a general schematic to give you the sense that the testing that was done, the PCR testing to confirm infection, was all prompted by symptoms. So there was no systematic testing of individuals who were not symptomatic, except for the collection of bloods periodically throughout the course of follow-up to allow for an assessment of seroconversion. And that's the way that the studies, to some extent, provide some information on acquisition of infection, regardless of symptoms. But a key consideration early on was, what should the primary endpoint of these trials be? And you can think of the endpoints, the potential endpoints, as in this diagram, where at the top level there's sort of acquisition of infection, and then some of the individuals who acquire infection become symptomatic, develop COVID, and others are asymptomatic. And then among those who are symptomatic, some develop severe disease and mortality. Others have experienced non-severe disease. And so at first blush, it may appear that there would be a strong rationale for targeting prevention of infection. That would be the best effect that a vaccine might have. It could be said. However, experience with prior pathogens, and especially respiratory infections, had shown that that vaccines often do not have high efficacy at prevention of infection, and rather have much greater efficacy at preventing disease and more severe forms of disease. So there was not a strong hypothesis that these vaccines would have high efficacy at prevention of infection. At the other end of the spectrum, clearly the prevention of severe disease is the most clinically relevant endpoint to the individual and to the public health system. But it was anticipated that severe disease endpoints would be exceptionally rare in these clinical trial populations, and that would not be viable as a primary endpoint in and of itself. And so it's in some sense as a compromise, and also consistent with prior studies of respiratory pathogens, a pretty classical design and primary endpoint was selected. That is the prevention of symptomatic disease, COVID-19 disease in the middle there. But all these trials collected all the endpoints that you see on this slide and are included in key secondary analyses, where, as I mentioned, the SARS-CoV-2 infection events that are captured are those that are seroconverting, not those that are PCR positive but do not seroconvert. So it's sort of a limited read on SARS-CoV-2 infection. The primary analyses targeted estimation of vaccine efficacy, which is classically measured as 1 minus the hazard of the endpoint in the vaccine versus placebo groups. As Stefan mentioned, many of these trials used a Cox proportional hazards model to estimate this parameter. Used exact binomial methods or other methods, which tend to produce highly consistent results in the context of rare endpoints, and that has been validated in longer term follow-up of these data. Most of the studies did conduct their primary analysis in a subset of the trial participants, namely participants who were retrospectively determined to have been SARS-CoV-2 negative at enrollment, and also only counting events that occurred a certain lag time, T or more days post the last vaccine dose. And this is a common technique in vaccine studies to allow for the full effects of the vaccine to manifest before endpoints start to be counted. It was a common and required strategy that each of these studies also did analyses in the full enrolled population in an ITT sense. The success criteria were laid out early on by the US FDA and the WHO, and very helpfully were in agreement with one another, and stipulated that a successful vaccine would have a point estimate for vaccine efficacy of above 50% and the lower bound on the 95% confidence interval of above 30%. So this success criteria really guides the required size of the trial to ensure that under a given design alternative, success could be established with high probability. And so specifically, these were all event-driven trials, and one can calculate that under a design alternative of 60%, which was the design alternative used by most of these studies, surprising given how effective these vaccines have been seen to be in reality. So under a 60% design alternative, one can calculate that 150 or so primary endpoint events are needed to have 90% power to establish success using this criterion under a 2-to-1 vaccine to placebo allocation. And so then we work backwards to of course identify the total sample size that's required by specifying the proportion of individuals who would be in the primary analysis cohort and the underlying incidence of COVID-19 in the SARS-CoV-2 negative placebo group to calculate how many participants need to be enrolled. And so, for example, this 30,000 participant number was derived based on an assumption of a 1% 6-month placebo incidence, which, as Stefan mentioned, turned out to be quite a conservative assumption as to placebo incidence in some of these studies. Time was really the key commodity in designing these trials, and it was important that these trials were large enough such that under reasonable assumptions as to incidence and vaccine efficacy, that the primary event-driven analysis would occur within at least 6 months of trial opening. Furthermore, it was important that the first interim analysis for efficacy would occur by at least month 4. And for most of these trials, that occurred when 50% of the target number of primary endpoint events had accrued. These were, in retrospect, really key design elements. And as we know, each of these studies essentially established efficacy at the time of the first interim analysis, well before 6 months of trial opening. Okay, so this is a high-level snapshot of the primary efficacy results for each of the trials that have been conducted in the U.S. I've added the Pfizer trial, which did not receive U.S. government funding here, but is a licensed vaccine in the U.S. And in blue are the studies that we've been involved with under the co-VPN. I'll just highlight a few key conclusions that can be drawn from these data. One is that there has been, generally speaking, remarkably high and definitive efficacy established for each of these vaccines, especially for the mRNA vaccines. They're at the top with remarkably high 95% efficacy during the primary analysis period. Another feature of these studies is you can see how little follow-up had accrued by the time of the interim analysis across each of these studies, with about 1.5 to 3 months follow-up on average having accrued post-second vaccination by the time of the primary analysis. So these trials reported out early, and really this raises the mandate for continued follow-up of these participants to fully establish longer-term safety and efficacy. Another note is that generally speaking, as was anticipated, we've seen higher efficacy at preventing more severe forms of disease than less severe forms of disease. Although, as shown in the right-hand column of the slide, as was expected, the incidence of severe disease was quite low in these trial populations, and so, especially based on the primary analysis, there's not great precision around what the efficacy is against severe disease, again, requiring further follow-up for a more definitive evaluation. Not shown on this slide, we've also seen quite remarkably consistent results across various demographic and at-risk subgroups. This was a concern, given that some vaccines do have attenuated efficacy in older populations and populations and other populations. This has not yet been seen for these vaccines. And what's not shown on this slide, but what has been seen as these results roll out and outside these trials is there is evidence of heterogeneity against in protection across various geographic regions, very likely due to different circulating variants that began to emerge as these results were rolling out. And so digging into that a bit more in a couple slides. So first, what I'll comment on is how these trial designs were adapted given the early efficacy results that were established here. There was considerable discussion once these trials began to read out as to how the trial designs should be handled and how the placebo recipients of the trials should be treated, whether or not they should continue to be followed under placebo or not. And after a good deal of discussion, it was determined that both for ethical as well as for practical reasons, in terms of the imperative to retain these participants on the studies, given that the vaccines were becoming available outside the studies, it was determined that the best approach would be to offer the vaccine to the originally randomized placebo recipients under what was called a deferred vaccination approach. And this design and the statistical considerations around how to analyze data from this design was really led by Dean Follman and his colleagues at NIAID in collaboration with a large group of individuals at the CoVPN. This is a schematic just showing what this design looks like, where individuals on the placebo arm, once efficacy is established, are offered the vaccine, and everyone continues follow-up. Ideally, this is done in a blinded fashion where the individuals originally randomized to vaccine are offered a placebo at that point in time. Some of the studies have done it in a blinded fashion. Others have taken a more practical approach in doing it in an open label fashion. And while at first blush, one might be quite concerned about how to evaluate longer-term safety and efficacy under this deferred vaccination approach, what was shown is that under certain assumptions, longer-term safety and efficacy can still be evaluated quite rigorously by exploiting the randomized trial design. And this is a graphic that attempts to illustrate the concept here, which is that under this hypothetical data here for a trial that has a blinded follow-up period from August to February, where let's suppose that the COVID event split that's observed in the vaccine and placebo arms is a 25 to 125 split, resulting in a point estimate for vaccine efficacy of 80% during the blinded follow-up period. If then after crossover of the placebo recipients, there's a case split seen in the original vaccine group versus the deferred or crossed over placebo recipients of 60-30. If one can make a constancy assumption, namely that the short-term benefit of vaccination to the newly vaccinated participants is the same as to the original vaccine recipients, an 80% reduction in disease, one could then infer that there would have been 150 COVID events in the post-crossover period had a placebo arm continued to have been followed. And with that inferred or counterfactual placebo, one can then evaluate what the later vaccine efficacy is by comparing the COVID event rate in the original vaccinees to this counterfactual placebo, and see in this hypothetical example that the original level of 80% efficacy had declined to 60% in the second period. And this is a conceptual illustration. The estimation can be done with a time-varying Cox model, and of course generalizes to multiple periods and continuous time. But there's a key assumption underlying this estimation, which is this constancy of the short-term effect of vaccination over time. And while initially that assumption looked entirely reasonable, as new variants of the virus began to emerge, this assumption became highly questionable. And really has raised the imperative for extension of this methodology or new methodology to evaluate longer-term efficacy in the context of these emerging variants. So this is what the data in the U.S. has looked like in terms of these emerging variants. This is data from this is a beautiful graphic from the NextStrain platform showing over calendar time the proportion of circulating variants that are by circulating viruses by variant, and underlaid below that are the 4 trials with results so far. And we can see that the blinded and placebo-controlled follow-up period from the trials really doesn't extend into the period where we have these variants of concern emerging in the U.S. And so if the vaccine is differentially protective against disease due to different variants, this really compromises the constancy assumption, because it's quite likely that the short-term benefit of vaccination post-crossover may be different than the short-term benefit of vaccination pre-crossover. And we need additional methods and external data to reliably evaluate variant-specific vaccine efficacy and how that efficacy varies over time. So I'll show you conceptually one approach to doing this, and this is to leverage surveillance data to evaluate a variant-specific vaccine efficacy and how that varies over time. So again, I'll take my hypothetical example here where I have a trial that has a COVID event rate split of 25 to 125 in the blinded follow-up period. And then the concept here is that we have what we call a modeled placebo. So we leverage broad surveillance data of detected infections, hospitalizations, and deaths, and an existing population model of COVID-19 incidence to characterize incidence among the unvaccinated. And this model is localized to the particular geographic regions where we have trial sites. And then it's even furthermore tried to make specific to the trial cohort by adjusting that incidence estimate upward or downward according to the individual trial characteristics of the trial population, namely age, race, comorbidities, and baseline SARS-CoV-2 infection status. So suppose in this hypothetical example that the modeled placebo would have predicted that there would be 150 COVID events during this blinded follow-up period. So given this modeled placebo, what we can then do is to estimate what we call an offset, which is how discrepant the model is from the observed placebo during the blinded follow-up period. In this hypothetical example, we estimate that the offset is 20%, that the observed placebo incidence is 20% lower than would be predicted based on the model. Then we make a constancy assumption, a different type of constancy assumption. Having observed in the post-crossover period the number of COVID events in the original vaccine group and the deferred vaccination group by variant, and here I'm showing it by pre-Delta variants and Delta variants, if we can make an assumption that this offset is constant over time, and there's some data to evaluate that assumption during the blinded follow-up period. And furthermore, if we can make that assumption that offset is not differential by variant, then we can infer the counterfactual placebo number of COVID events by variant in the post-crossover period. And so, for example, for the pre-Delta variants, in the middle column of the slide, we can infer that the original vaccine efficacy of 85% had declined in period 2 to 43% against the pre-Delta variants. So the key here is that using the modeling, we can attempt to recover variant-specific counterfactual placebo and variant-specific vaccine efficacy and how that varies over time under this strong assumption of a constant offset. Because this is a strong assumption and there's only limited data to evaluate this assumption, we think it's really critical to triangulate the results from this type of method with other methods that make different types of constancy assumptions to really achieve a reliable inference. So this is just one approach that's being employed. I'll highlight a couple of open questions. I won't get into too much detail given that there's just a couple minutes left. One open question is to what extent these vaccines reduce transmission, either by blocking infection or by reducing transmission from an infected person to their contacts. There's considerable challenges with doing this, given the features of SARS-CoV-2, including the short duration of of infection and infectiousness that really require frequent and intensive and systematic testing to detect infections. A number of studies underway to interrogate this question, and really critical statistical challenges in addressing this question, including bias due to length bias when in the context of infrequent testing, confounding, as Stefan mentioned, both due to factors that influence exposure to virus as well as factors that influence transmission of virus to others. Misclassification error for studies that are actually attempting to identify transmission events, and disentangling vaccine effects on infection and secondary transmission. I'll just very briefly mention the very high penultimate question in vaccine development, which is the discovery of an immune correlate. That is a biomarker that predicts the level of vaccine efficacy. So this is the holy grail of any vaccine program, because having discovered an immunological correlate, one can then greatly accelerate the development and licensure of additional vaccines, as well as the licensure of vaccines for new populations. We can't do an efficacy trial in every single population, and we can't do an efficacy trial given each new update and variant version of a vaccine, or adding a new adjuvant to a vaccine, and so on. This is a strategy that's been pursued for many different vaccines, and outside vaccine development, it's really analogous to development of a surrogate endpoint. So there was a considerable pre-planning for this, including the construction of these harmonized protocols, harmonized immunological laboratories, and a common data statistical group to implement a variety of different statistical frameworks to interrogate this question in a systematic and consistent fashion across these protocols. And the first paper under this program has been published. And so we can talk about this more in the discussion if there's interest. I'll just summarize by really highlighting what I think is really evidence of strong success of the strategy that was taken here, which is pretty simple, classical, large designs to provide rapid results, definitive answers, and adapt the designs to the emerging data, which has resulted in now 3 licensed or approved vaccines in the US, more at a global level. But there remain really critical, open, challenging questions that are going to require not just data from these phase 3 trials, integrating the data from the phase 3 trials and considerable array of observational studies that are underway, and triangulating the results from multiple statistical frameworks, given the challenges with addressing these open questions. So I'll stop there.
Wow.
I think I'll stop sharing here.
All right, thank you very much, Holly. It's really interesting too. I think we get enough to discuss all the weekend, actually. So, all right. Anyway, okay. So Antoine started to ask questions in the question window, but as you're a member of the scientific community, Antoine, you can ask your question by yourself, with or without the mask. So you just have to switch on your mic and ask your question. So please. Go.
So my colleague is not in the office anymore, so I can take my mask off.
Right.
Thank you very much for the inspiring talks. I have 2 very naive questions. The first one is for Stephen. You showed something which was very interesting, where the time between 2 jabs makes a big difference in terms of the protection provided by the second jab. And I wonder, is there a way to to learn how to best balance the time between the 2 jabs in order to optimize the number of persons, the average number of persons who might potentially get infected?
Yeah, I think it's a good question. It's intriguing to think, could one do this? But there are a number of problems. First of all, what I showed you was figures for the antibodies, not actually protection. And it then depends upon what, if you like, assuming even that the antibodies indicate some degree of protection, whether it translates approximately linearly or how does it translate. It might well be that a much, much lower level of antibodies is as much protection as you're going to get, and you get hardly anything by going higher up. So it could be that the effect of a longer duration is exaggerated here and it doesn't make that much difference. But maybe not. It could be the reverse. So that's the first point. The second point is, okay, the information that I showed about the baseline values and after the first dose, which of course can't have been affected by the interval, is reassuring that the explanation is not due to different subjects having been involved, but we still can't be sure. It's not as if this was a randomized study. It's a finding as a result of an error. It's an error that AstraZeneca made. They gave the wrong dose, and then having given the wrong dose, they then had to try and sort things out. And this sorting things out time stretched the time for the people getting the second dose. So I just don't know. I don't know enough about the field in general to know whether there are other data which would help us. I think these data are not good enough on their own. They're suggestive, but they wouldn't actually help you to design an optimal program.
Yeah, I don't know whether Holly wants to comment also, because as she's very much involved in design of vaccine clinical trials. But I guess that it's really a good point, because it's also it could really change probably the efficacy of the vaccine, controlling the duration between the injection. But the issue is really also the timing of the design then to explore this. And yeah, I think it's a great point, but it's most often it's difficult to implement. I don't know, Holly, if you want to comment on this too.
I guess the only thing I would add to Stefan's response is that there is reason to be interested in this hypothesis, not just from the AZ data, but there are other vaccines where there has been rigorous evidence presented suggesting that there's higher efficacy with a longer interval between the two doses. So there's reason to wonder about this question. But I agree with Stefan. It's exceptionally difficult to evaluate this in the context of the efficacy trials alone, given that this wasn't a systematically interrogated question. In vaccine development in general, this is something that's typically evaluated in earlier phase research. But of course, the limitation there being that it's purely based on the immune responses that are generated and not based on the efficacy. It's quite rare that one would use an efficacy trial to systematically interrogate the efficacy associated with a shorter interval versus a longer interval, because obviously that requires a massively sized trial. So oftentimes we are stuck with this setting where we have to infer this based on observational data. Yeah, nothing more to add.
But I know that you get some questions too, so you want to ask them.
Yeah, thank you, Rodolphe. So first of all, thank you, Stefan and Holly, for very nice presentations, very interesting. I had a question for you, Holly, but also would like to get input from Stefan. You briefly mentioned that there was a thinking of doing a platform trial, but that probably due to the speed and the feasibility, it wasn't really the time to properly think about it. But suppose that for a future pandemic, you would have time to think and prepare. Would there be advantages? What would be the main challenges? Any considerations around that?
Yes, certainly statistical advantages associated with having a common control group and a common trial with which to evaluate each vaccine individually, as well as head-to-head efficacy of the different vaccines. And there is the WHO Solidarity trial underway, which did elect to take a platform approach. So it's not that it can't be done, but in the judgment of the particular partners in the U.S., it was determined that would not be an expedient way to conduct the trial, because typically the key disadvantage is time, the time that it takes to set up and procure agreements from multiple parties, multiple manufacturers to have a harmonized and common protocol. Yeah, so I would say it has great statistical advantages as well as resource advantages, in the sense that a common and single control group is used for each of the vaccines, so it requires a smaller sample size.
Okay.
So just one comment. Obviously, as we know from I think it's Dunnett's test, then the optimal design requires you to allocate more patients to the control group than to any other vaccine group. So we would actually be looking at the opposite of what was chosen for AstraZeneca, Oxford, and also for Novavax. There's also, I think that everybody here understands this, but some people get very excited because they think, oh yeah, and you can use the historical control patients as well, but you can't. You simply can't, because even for other disease indications you can't, but for efficacy in infectious diseases you simply can't, because the infection rates will vary so rapidly over time that you still have to stick to the principle of concurrent control. So you don't necessarily gain as much as one might think by doing this, but it certainly has some advantages.
Right, and just to clarify that the Solidarity trial and any platform trial that would have been considered would only have been using the data from concurrently enrolled and followed controls. But still feasible to do that, even in the context where some of these vaccines didn't become available. There was staggered entry of different arms. Still possible to do it with concurrent controls. But I agree with Stefan that the statistical advantages get lost as further spread out in time, the availability of those products become.
It's actually a little bit linked to this. I was wondering, Ollie, if you want to check whether your assumptions are fine when you are doing the crossover exercise, trying to estimate the long-term effect of the vaccine. So providing that at a given time we'll have some idea of any surrogates marker and so correlate of protection and probably neutralization, and providing that we start to have some estimates also of this surrogacy, then maybe it could be you can compare then what you get as an estimates from your approach to the one measuring the neutralization and trying to compare then your estimates on the long-term basis. I don't know if you have already started to do that, or maybe it's a bit too early. I don't know.
Yeah, so excellent. Yeah, so definitely a part of the correlates program is to evaluate the immune responses, their associations with efficacy, and then, as you say, one can evaluate the immune responses to different circulating variants to then infer, what efficacy would be predicted to be based on that immune response against different circulating variants, and that would be an interesting quantity to contrast with the efficacy that we estimate using the various different approaches to evaluating variant-specific vaccine efficacy. I would say basically that's underway as part of this correlates program. Of course, there are multiple sources of the evidence outside the phase 3 trials that I haven't really focused on that are trying to evaluate that same question.
Stefan, do you want to say something? No. Okay.
Well, just to say that one issue, I think, which has been realized in recent years through a lot of theoretical work, which has been done on using historical data to boost the precision and power of concurrent clinical trials, is that one has to be very, very careful that one doesn't overlook sources of variability. When these sources of variability are entered formally into the calculations, one sees that one has far fewer data in practice than one imagined. We were involved in a study, not in vaccines, but in another indication, where about 1,200 historical controls could be identified, but they were from a number of different studies. And when we looked at the studies, the study variation, we put it into the model, at best, the most optimistic assumption was it was like having 50 concurrent controls. So you thought you had a huge amount of information, and it went down to very little at all. And what worries me with a lot of epidemiological studies is that actually these sort of effects are not incorporated formally in the standard errors. One way of doing it with the vaccine studies, to attempt to do it, might be to include center effects in the model. And then you would actually force within center comparisons if they were fixed. If that happens, then immediately the data just disappear. You're left with almost nothing because people are being incorporated at different times. Or you can try optimistically to include as a random effect, and then in that case, your standard errors will increase. But instead, what people tend to do is they tend to say, well, once we've conditioned on all these covariates, let's pretend it was a parallel group trial. So basically, once everything is conditioned on, you then sort of imagine that it's perfectly well behaved, but in practice, they're not. And that's not a reason for not doing it. I'm just saying one has to be very, very cautious about things like standard errors, confidence intervals, and so forth.
I think I agree too. I don't know, Marie-Carole, do you have any question to raise? Because I'm sure you all know Frank Carrel, who is connected. And I must say that I'm very surprised not hearing any questions from him right now. So Frank.
Well, I have one.
Oh, you're here. Okay, all right. Good. I know that there was an issue with your mic. Okay, all right. So you can ask your question.
This is a very technical question for Stephen regarding the choice of prior. We know that in most drug development settings, we know more about relative efficacy than we do about the outcomes in individual treatment arms. And with a binary outcome, one of the most popular choices of priors would be on the log odds ratio scale, say using a normal normal prior for log odds ratio. Would you have the same concern about that as you had for your concern in these in that vaccine trial?
I think that the theta scale that was used is very similar to the log odds ratio scale. I did do some work at one point and trying to see what the transformation was. I have it somewhere, but I came to the conclusion that I might as well just analyze on this theta scale, which is essentially the relative risk reduction scale. And that has some advantages for the following reason. You can actually say that most of the individuals in the clinical trial are irrelevant because they're not going to get COVID. And since they're not going to get COVID, giving them a vaccine is not going to protect them from getting COVID. So the ideal trial would only enroll individuals who are certainly going to get COVID. And you would then have a look at them under placebo, and you then have a look at them under the vaccine. Now, the estimate of how many such individuals there were is actually given by the number who were infected in the placebo group. They're essentially people who came into contact with the virus in a meaningful way, in a way that gave them infection. So that's the correct divisor to use. And so here, for this sort of particular analysis, I think the relative risk reduction scale is a good scale to use. And one could be optimistic that it might be fairly constant, irrespective of the level of the pandemic at the time that the trial is run. I don't expect it to be perfectly stable, but fairly stable. So that's fine as long as you observe concurrent control. As soon as you depart from concurrent control, then in that case, if you start mixing placebo cases from elsewhere and vaccine cases from one place, then you have a problem. But as long as you've got concurrent control with that particular scale, I think things are generally fairly good.
And I have a hopefully quick last question. In Bayesian methods, there's no formal role for a sample size calculation. Would it be fair to say that sample size calculation, if you're going to use a Bayesian design, just slows down the launch of a study?
Well, yeah, I'm not sure. I mean, there's been a lot of work, modern work on the value of information. And that's the way in which I see it. The basic, and you can even look at a frequentist calculation this way. If you look at a standard calculation of I don't know, 90% power and 5% significance, two-sided, although it's really one-sided, 2.5%, you're talking about, or 80% power, you're talking about a signal to noise ratio targeted from the data of about 3, something like that, 2.8 to something a bit over 3. And that, I think, can also have a Bayesian justification. That's the amount of information you want to have something useful to say. And I certainly think, and Holly talked about this, she mentioned explicitly the 150 target number of cases which came out. I think you could justify that in a Bayesian way as well. The point about it is that you want to be able to have enough information for it to be valuable to take a decision rather than collecting more information. That's the point at which the trial should stop. And you could justify that. The 30% minimum, it was a mistake of Pfizer to put that in the prior. It doesn't belong in the prior. That's an objective. You're trying to prove that you have at least 30% efficacy. You could either do something fancy with a loss function, or you do it with, a targeted posterior probability being 95% that it's above 30% or something like that. But to have it as the prior distribution was completely wrong thinking and non-Bayesian, but didn't matter because, it was so uninformative anyway. Who cares?
There is a. Okay, sorry, Antoine. Go ahead. It's fine.
Thank you very much. I have a question, a very general question. So the pandemics had a huge impact on our lives. You just have to think as an example about the way we work. I mean, working remotely has become almost the standard, which is quite amazing if you think about the state of the affairs 2 years ago. And I wonder, I mean, did this rather fast randomized clinical trials that were made, especially for this to respond to the COVID-19 pandemics, will that have an impact on how we carry out RCTs in the future?
Well, Holly's the person who's actually done some work. I've just been a parasitical observer on what's been going on. So I'll leave her to answer this.
You can just correct anything I say, please. I guess I'd love to think it would have a benefit. I do know that there was considerable effort invested by many, probably including many on this call, following the recent Ebola epidemic to develop what were termed sort of I'm missing the term, template or sort of pre-planned protocols in the event of another pandemic, and to expedite the implementation of vaccine trials. I guess I'm not personally convinced that had a huge benefit on the speed with which the COVID vaccine trials were implemented. There are so many considerations that are specific to a given pathogen, to a given pandemic, to the data that are available at the time, to the extent of development of the assays that are required to diagnose infection. So many specific considerations that make development of kind of an abstract protocol difficult. Nonetheless, there are some themes, I think, here, from COVID that certainly the framework and the kind of consensus around the deferred vaccination approach, I think will certainly have application beyond COVID, well beyond COVID. But that doesn't necessarily expedite the initial development, ramp up, and implementation of a protocol. Yeah, so I guess I'm not terribly, but I don't see a lot of ways in which this will benefit us. Clearly, COVID was benefited tremendously by the massive investment in development of other vaccines, especially and most especially HIV vaccine development, especially in the US. There had been so much money and infrastructure and research programs set up to develop HIV vaccines, and all those people were immediately pivoted to COVID and the networks that had been established for HIV, and the laboratory statistical centers, clinical trials networks, and sites were immediately pivoted for that application. So that has a direct and immediate benefit. Stefan, what is your sense?
I think this is true. I mean, it's difficult for me to judge to what extent remote working and so forth was a benefit or a problem in the development of the vaccines, but I think the response was magnificent. To answer Frank's question, the main effect of the pandemic has been the ratio of beer that I consume at home to beer that I consume in the pub has increased considerably. Luckily, I have a supplier who will actually ship the beer to me to my home door. But yeah, we've lost a lot of things in our life, but I honestly not so sure that meeting face to face has been the most serious one that we've lost. I think there's a social aspect to that which is important, and maybe there's a long-term effect on work as well. But I think in the short term, we've managed to work fairly effectively remotely. I don't know whether that's what Frank is getting at.
At least, so just to complete, so that's why we are doing an experiment by organizing this webinar now and having a physical conference in September, and then we could compare how it fits in these situations. All right. And so just if I can, I'd like to come back to one technical point, because it's not technical, but it's one of the issues that actually you both mentioned, is the unbalanced allocation done by Astra and Oxford. And I think what is interesting also is that Holly mentioned some of the arguments to do this. And especially also, when you look at longer term effects, having some placebo to do that. And Stefan, you clearly demonstrated one of the potential limitation of this. So then my question would be, would you do the next time, would you do a trial with unbalanced allocation finally, or you go back to a balanced allocation?
Well, Holly raised an interesting point, and that is because the measure is non-linear, the minimum variance allocation is not one-to-one. But I had a look at this and I came to the conclusion that 2-to-1 was actually worse than 1-to-1 as regards that. But 1-to-1 would not be the optimal. But that's only part of the story. Part of the story is also if you're monitoring for the purpose of stopping at a particular total number of cases reached, then in that case, that particular total will be reached later if you use anything but a 1-to-1 randomization if the treatment is efficacious. And since speed was so important, I happen to think, but I mean, I can see there are counter arguments. I happen to think that the AstraZeneca-Oxford choice was not a good one. I was interested to see that Holly also mentioned, I think, Novavax. That must be the US trial, I think, because in the UK, they went for a 1-to-1 randomization. No. There are some advantages. Sometimes it's claimed, well, we're interested in side effects, and so as many people who are vaccinated as possible. But there again, that assumes that you're looking for rare side effects. I think for common side effects, you really want a placebo control group to judge whether they really are due to the vaccine or not. And therefore, you're not necessarily better off with a 2-to-1 randomization. But for very, very rare things, yeah, it might well be to have more people treated with the vaccine.
Yeah, I guess all I would add is that at this point in time, I'm thankful that the U.S. AZ trial did use a 2-to-1 randomization ratio because they were the one study that did not take up this approach of deferred vaccination, and so the placebo recipients from the AZ trial have largely been lost in the sense that they're no longer contributing information to the study. And so the vaccine group is all that more valuable. And I'm grateful that we have twice as many of them, as placebo recipients. So yeah, hard to answer in generality. But in this particular instance, I'm grateful that we still have more participants remaining under follow-up, so we can evaluate these longer-term vaccine effects and against different variants.
All right. So I don't know, is there any last questions that one member of the scientific committee want to ask, or we're done? All right. So yeah, I'd like to thanks very much. So first of all, both of you, Stefan and Holly, for your outstanding presentation. It was really fascinating, and so thank you very much again. I'd like to thank also the scientific committee for giving a hand in organizing this webinar, and especially also I'd like to thank Sandrine Fougère, who managed all the organization and the technical aspects of this webinar. There is a poll that if you can answer the question, it would be nice to see what you think about the next conference. And last but not least, I'd like to thank also our sponsors that will help us to organize the physical meeting that hopefully we will organize in Paris in September. So thank you again, everybody, for participating to this webinar, and so have a good evening, good day also for those who are at the other side of the world, and have a good weekend too. Thank you very much, and see you very soon.
Thank you very much.
Bye.
Thank you.
Thank you all.